uniform discharge is initiated, and in the discharge current waveform, we will see a single peak per half cycle of the applied voltage. That is the indication of uniform discharge. This is the spectroscopic method through which we measure the plasma properties like density and temperature of electron. So this is generally we use a collisional radiative model. This is using the helium line intensity ratio, and we just by comparing these line ratios, we are able to find out the density and temperature. So in our case, we are having the density in the range of 10 to the power 11 per cc and temperature in few electron volts. And this is again the indication of non-thermal cold plasma. Then we explored application of the plasma, the DVD plasma in the plasma stealth technology. In the stealth technology, basically we can have a plasma layer that is created on a flying object, so that the waves, the microwaves coming from the radar cannot observe the object, and they cannot identify that there is any fighter plane available nearby or not. So this is dependent on the four parameters that the microwave attenuation. So one is the plasma frequency, the collisional frequency of electron neutral, then the microwave launching frequency, and the plasma thickness. So what we have generally started this work with a parallel plate electrode, because we are not having a very thick plasma initially, so that we can identify a large attenuation. So what we did the experiment in the parallel plate electrodes, and we launched the microwave from this side, and it propagates along this length of around 10 to 15 centimeter, and it is received by a detector. So we will be able to obtain that in microwave attenuation of around 0.3 dB in our experiments, and then we have compared the density coming out of this attenuation with the spectroscopic observation, and that matches very well. So this experiment was wonderful, and we have obtained that whatever the plasma parameters that we are having in our experiments, the corresponding attenuation we are able to achieve. But certainly, if you want a more attenuation of microwaves, then you have to create a thick plasma on a surface of it, on the aircraft or somewhere else. But somehow this technique is also used as a diagnostic method to find out the plasma density over here. Then we switch to the planar DVD plasma, because we want to create a plasma over a surface. So we use this type of electrode system over here, that this is a dielectric sheet, and then we mounted the electrode like a connected spirals. Then you can see this is a E-state electric field simulation. So between the electrodes, the electric field is very strong, but when we move away, the field diminishes. That you can also observe in this discharge current waveform, that initially it is very slow, but finally it goes a sharp peak over here. So we are able to create this type of planar plasma sheet on a surface of, but certainly we have created, but the plasma thickness was not very great. So that we have also done the experiment on micro attenuation later, but not for the plasma stealth point of view. We have also observed some of the properties like a plasma spot formation, regular patterning on the planar plasma sheet. So for a lower voltage, we are able to observe that the plasma spot pattern is initiated, that we just reduce the voltage from 2 kV to 1.4 kV. The uniform plasma converts to the plasma spots because of the uneven charge distribution on the dielectric surface. Then we have also studied the ionization waves in the planar surface DVD, and we were able to observe that there is the initiation of the negative wave with a displacement dispersion relation of omega k is equal to constant type of relation. So this is the first time we have observed in our experiment that there is the stripe fluctuations in the form of the ionization waves. Then we have also obtained the square lattice pattern in the planar DVD plasma by just changing the frequency of applied voltage. So if I reduce the frequency from 10 kilohertz to go to the 2 kilohertz, then the random plasma spots are converted to a square lattice, and we have analyzed all the things over here, and the perfect lattice was observed. We did the application of microwave attenuation over here in the planar plasma also, but the attenuation that we are able to observe over is only 0.03 dB because the plasma thickness was very small over here. So, but this is actually established as a very good diagnostic method to obtain the plasma density by the microwave attenuation measurement. So this was a paper published in 2023. Then the third geometry that we have done the work on this DVD is a coplanar DVD plasma, and this is basically the plasma display cell or the plasma television, where this is basically a 3 electrode device, and the plasma is produced between the 2 electrodes. Like this is here the same plane, and the 2 electrodes are mounted in the same plane. So when the potential is applied between these two, the discharge is ignited over here, and the UV radiation that are emitted from the plasma is interacting with the phosphor layers over here to produce the white light. There's a combination of RGB, and we are able to obtain the color grades or the variations based on the different signals that were from the 3rd electrode. So this is how we are able to get the micro discharge in the plasma panel, in the plasma display panels. The gases that we use over here is the xenon and neon combination. The wavelength that we obtain for the UV is 147 nanometer and 173. We have done a lot of different configurations and devices for the micro DVD plasmas. So they are basically all applied for the patents: lifted electrode structure, single plate electrode structure, fence electrode, and groove dielectric structures. So major approaches that we obtain are the optimized cell capacitance, long discharge gap, face-to-face discharge, and cost. And cost is basically a very important factor over here. So that leads to the high efficiency of the discharge in the plasma display panels. Finally, the 4th configuration that we did is the coaxial DVD plasma, that is in the form of plasma jet. Here in the plasma jet, we are having the single pin electrode and also one electrode that is in the form of a ring. So when you use a flowing gas over here and apply a potential between the electrodes, then generally there is a spray of the discharge which is coming out in the form of plasma plume that we treat as a. And this plasma jet is proposed for the biomedical applications like skin, dental treatment, cancerous cells, wound healing, sterilization, etc. And finally in the agriculture for the seed growth, germination, nitrogen fixation, material processing, food sterilization, etc. So they are the prominent applications of the plasma jet that the people are using nowadays. There are different configurations of plasma jet that are reported from various papers. In our experiment, we have also produced the plasma jet by this pin to ring electrode configuration, and these are the discharge current and voltage diagram, where you can see that in the case of helium, we will able to see a single peak, but in the case of argon, there is a multiple peaks in the current that is more filamentary nature over here. These are the pictures of the helium and the argon. In the case of helium, the plume is very uniform, but in the case of argon, it is you can say it like a turbulent type of stream is obtained. You can see if I increase the voltage, you can just consider 0.4 kV to 4.6 kV. There is a linear increase in the length of the plasma pool. We will able to get 28.5 mm of the length at 4.6 kV in the helium. But it is not in the case of argon. In the case of argon, initially the length increases and again it decreases. Also, the discharge quickly converts to the turbulent mode, or you can say the chaotic mode, if you just increase the gas flow rate or increase the potential. But there is you can say the game between these two. The helium is a costly gas. Argon is is having a low cost. So generally people use argon because the helium, managing the helium at regular experiments is very, very difficult. This is the Reynolds number analysis for giving the laminar and turbulent modes in the plasma jet. So in the case of helium, the value is very, very less in all the range of the flow rates. So it always remains laminar. But in the case of argon, for some cases, it remains laminar. And again, it going to the turbulent mode for higher flow rates. We have also measured the plume temperature and its variation along the length of this plume. So that having at a tip, we are having a maximum temperature and then it reduces as we move away from the tip. But in the case of helium, it again increases after some time because of the thermal conductivity of helium. We have also did some experiments on the plasma dielectric interaction using the plasma jet in order to get the plasma processing application, some of the basic studies, and we are trying to get something. We have also measured the temperature of the dielectric after the plasma treatment, and it follows that the trend T is equal to T naught e to the power minus kx type of variation. From plasma medicine, different people are working in different labs across the world, and the major part that is played over here is the reactive species ROS and RNS, and a lot of air plasma chemistry works because the discharge interact with the ambient air after coming out of the jet. So there are also a few of the applications where the UV radiation impact different type of. So major is the germicidal wavelength. This is 240 to 280 nanometer that is produced in the plasma by the combination of the gases over here, and a lot more applications with the different other wavelengths in the UV range. At BIT Jaipur, we have produced the plasma jet in the helium and argon by using this type of setup, and we are trying to obtain some of the prominent applications like plasma activated water and the biomedical applications. We have also tested the E. coli bacterial growth by using our helium and argon plasma, and we are able to get that the most of the E. coli is not growing after the treatment. But the argon gas is giving more effect as compared to the helium. There's a quick interactions in the case of. There are the few events that we have organized at our campus. In 2021, we have organized the 36th National Symposium on Plasma Science and Technology. Then in 2022, we have organized the Plasma Gyan. This is basically the event for the school and college students about the plasma, the awareness of the plasma technology. Then recently, last year, we have organized the international workshop on cold plasma technology and applications during 6 to 8 February. And there is a very nice presentations and from the worldwide. So thank you very much for your kind attention. We are celebrating our 70 now the 72 year presently the Platinum Jubilee celebration for BIT Ms. Ranchi. So, any question if someone want to ask? Yeah. Thank you. Okay. Thank you. Thank you.
Thank you. Thank you, sir. Is my presentation visible? Okay, so good morning, respected colleagues, distinguished delegates, and students. I'm Dr. Jaipur, Professor at Banaras Hindu University. It's an absolute privilege for me to welcome you all to this conference session that is focused on silent carriers, understanding the epidemiology of seed-borne plant pathogens. In this session, we'll explore the microscopic yet profound threats that seed-borne diseases pose to global agriculture and food security, when plant biosecurity and international trade. Is my slide moving? Can someone confirm?
Yes, it is moving.
Yeah. Let's begin by looking at the scale of global seed movement. Today's agricultural economy relies heavily on the international movement of high-value seeds, especially vegetables, ornamentals, and hybrid crops. However, as trade borders become more porous, seeds inadvertently act as primary vectors for phytopathogenic microorganisms. When an infected seed lot travels across continents and crosses the geographical boundaries, it carries the potential to introduce novel and new pathogens into pristine agricultural ecosystems, and that may lead to sudden disease outbreaks, severe yield losses, and monumental biosecurity costs. To systematically analyze seed-borne threats, we must ground our discussion in core epidemiological principles. Plant epidemiology, specifically known as plant epiphytology. It examines how disease incidence, prevalence, and spread evolve within plant populations across space and time. At the heart of this discipline is the classic disease triangle: the interaction among susceptible host, a virulent pathogen, and a favorable environment over a certain period of time. In seed pathology, the seed itself serves as the primary inoculum reservoir, initiating the infection cycle right at the moment of planting. What exactly defines a seed-borne pathogen? Scientifically, it refers to an infectious agent. It may be fungi, bacteria, viruses, or nematodes that are associated with seeds that retains the potential to cause the disease. There can be surface contaminants that are external to the seeds, or maybe deep-seated in tissue infections internal to the seed. The direct consequences are devastating, including reduced germination, pre and post-emergence damping off, and yield reductions ranging from 15 to as much as 90%. Furthermore, seed contamination introduces mycotoxins like aflatoxins into the food supply, posing severe public health hazards. So understanding our current diagnostic tools requires appreciating the history of seed pathology. The discipline began in earnest with Frederick Knopf's publication of Handbush der Samenkunde in 1876 and the founding of International Seed Testing Association in 1924. The mid-20th century was defined by Paul Neergaard, the father of seed pathology, who standardized several methods, including blotter and agar testing methods, for detection of various seed-borne fungi and bacteria. By the year of 1980s and 90s, the field underwent a molecular revolution with RISA and PCR being integrated with this detection of plant pathogenic microorganisms in seeds that enabled unprecedented diagnostic sensitivity and specificity to the procedures. The economic matrices surrounding seed-borne diseases are staggering. On a global trade, if you see, global scale, crop diseases cause an estimated 20 to 40% loss every year, translating to approximately more than $220 billion in direct losses. As illustrated on this slide, staple food crops like rice and wheat, they face catastrophic yield reductions during severe outbreaks. Beyond direct crop destruction, farmers face secondary expenses in chemical treatments, replanting costs, and export rejections due to strict quarantine barriers by importing countries. Coming to the importance of seed health, ensuring seed health is literally the foundation of sustainable agriculture. Healthy and pathogen-free seeds, they guarantee high germination rates and uniform seedling establishment, optimum plant population, optimizing water and fertilizer utilization. From a plant protection perspective, certified clean seed is our primary line of defense, preventing disease introduction rather than relying on reactive chemical sprays that we may have to do later in the season. It's also mandatory for meeting ISTA phytosanitary standards for international trades. As we look to the future, 4 major emerging challenges threaten seed biosecurity. First, climate change is altering pathogen geographical ranges and accelerating outbreak severity. Second, overuse of fungicides and bactericides has led to antimicrobial resistance among pathogens, rendering traditional treatments ineffective. Third, complex global supply chains facilitate long-distance spread of novel pathogens. And finally, the fourth, aggressive chemical seed treatments risk disrupting the natural seed microbiome and thereby weakening the crop's innate defenses. Before moving forward, let's clarify some key terminology. First, seed-borne means a pathogen that is present on or in the seed, whereas seed transmitted means the pathogen successfully passing from seed to the next plant generation to cause disease. Second, vertical transmission occurs from parent to offspring via seed, while horizontal transmission refers to the environmental spread between neighboring plants in the same season. We must also recognize bacterial biofilms and quorum sensing, which regulate initial tissue colonization. We now transition to the second section, epidemiological foundations. In this section, we'll dissect the biological mechanisms that allow seed-borne pathogens to survive inside dry seeds, activate during germination, and fuel exponential disease epidemics across entire agricultural landscapes. Revisiting the disease triangle in detail, an epidemic occurs only when all three components align over a certain period of time. So time has been considered to the as a fourth component, and this triangle is now being translated as a pyramid. So host factors include genetic susceptibility and growth stage. Pathogen factors, they center on inoculum density and virulence, where seed-borne pathogens provide the initial seed money for infection. And environmental factors such as soil moisture and storage temperatures, they dictate whether the pathogen remains latent or explodes into active disease upon planting. How do pathogens survive for years inside dormant desiccated seeds that have very low water content? They utilize, in fact, specialized survival mechanisms. Fungi from thick-walled spores or sclerotia, bacteria enter metabolic dormancy or form protective biofilms. Xerophilic capabilities allow survival under extremely low water activity. Furthermore, pathogens nest within deep micro niches, such as inner seed coat or embryonic tissue, protecting them from surface chemical dressings and UV radiation and whatever treatment we are applying externally. Pathogen latency makes seeds the ultimate Trojan horses of agriculture. During seed storage and transport, the pathogen exhibits zero metabolic activity and produces no visible symptoms. However, as soon as the seed imbibes water during planting, cell signals trigger pathogen germination alongside the seedling. This synchronized activation ensures the pathogen is perfectly positioned to infect tender seedling tissues as it emerges. Now, inoculum dynamics dictate how epidemics are built. Primary inoculum originates from the seed-borne propagules or crop residues and causes the initial scattered infection in the field for the first time in the season. Secondary inoculum consists of spores or bacterial ooze produced on these primary infected plants, which then spread rapidly via wind, rain, and insect vectors and are the main causes for epidemics. Management of primary inoculum requires exclusion and seed treatment, whereas secondary inoculum control relies on foliar spray and field sanitation. Even a tiny percentage of seed infection can trigger a devastating regional epidemic. A seed lot with just 0.1% of infection means 100 infected plants per hectare. So under favorable conditions, these 100 foci produce billions of secondary spores infecting neighboring fields across entire countries. Now, this exponential amplification highlights why zero tolerance thresholds are enforced by major seed-borne pathogens. The present slide illustrates the complete disease cycle. Stage 1, the primary inoculum resides in the seed coat or embryo. Stage 2, germination leads to systemic seedling infection. Stage 3, the mature plant expresses symptoms, producing secondary spores that infect neighboring crops. Stage 4, pathogens invade floral structures or developing pods, infecting the next generation of seeds. Interventions must occur at every stage, starting with seed treatments, followed by field roguing, post-harvest management. Graphing disease severity over time yields the classic model polycyclic disease curve. Now, in this and lag phase, we can see seed-borne primary inoculum causes initial scattered infection during sowing and emergence. In the log phase, secondary spread drives exponential disease growth during vegetative and flowering stages. Finally, the stationary phase, host tissue exhaustion or drying weather limits further spread of the pathogen, and controlling seed health minimizes primary inoculum, flattening the entire epidemic curve. So epidemics fall into two main mathematical categories, monocyclic and polycyclic, we all know. Monocyclic diseases such as cereal smuts complete only one infection cycle per season. Their severity depends entirely on the initial seed inoculum level. And polycyclic diseases such as leaf blights and bacterial spots repeat multiple infection cycles per season. They have cycles over cycles in a single season. And in a very little time, they have the capability and potential to devastate the entire field. For monocyclic pathogens, seed treatments provide nearly 100% protection or control. Whereas polycyclic pathogens, they require both clean seeds as well as seasonal field monitoring and treatment. Vertical transmission represents the ultimate biological strategy for pathogen persistence. In this process, The pathogen systematically invades the parent plant's vascular network, growing directly into developing ovules, gametes, or embryos. Research in rice seed microbiomes shows that dozens of core bacterial and fungal species are vertically transmitted across generations, ensuring continuous survival without needing external environmental reservoirs. Conversely, horizontal transmission occurs when initially healthy seeds become contaminated from external sources during crop development or harvest. Airborne fungal spores can land on mature pods. Bacterial ooze can spread via rain splash onto open flowers, or insect vectors like aphids and thrips. They can transmit viruses during feeding. Contaminated threshing machinery and storage containers also cause widespread horizontal seed lot contamination. Covered smut of barley is one of the major examples. Pathogens colonize seeds via 4 primary routes: direct floral entry through flowering, like a Stylogotrypsi; systemic vascular transport through the funiculus, seen in the Xanthomonas campestris; external surface contamination during harvest, as occurs in Tilacea caries; and indirect mechanical entry through wounds or natural openings, as is exemplified by infection from Fusarium moniliforme. Understanding these entry mechanisms dictates precise seed treatment strategies. It is vital to remember that seeds are not sterile vessels. They host a vibrant, complex microbial ecosystem, a microbiome known as seed microbiome. Vertically transmitted core taxa, both bacteria and fungi, colonize the seed coat and inner tissues. Many of these microbes are beneficial endophytes that promote seedling vigor and produce antipathogenic compounds. Modern seed pathology aims to protect these beneficial taxa while eliminating seed-borne pathogens. Seed germination represents the most critical window for disease establishment. During imbibition of water, seeds exudate sugars, amino acids, and organic acids into the surrounding soil. And these exudates, they trigger dormant pathogens, spores, and bacteria lying by, attracting them towards the germinating radicle. Weak or low vigor seeds remain in the vulnerable transition phase longer, dramatically increasing their risk of fatal seedling blight and damping off. The incubation period, the time elapsed between pathogen infection and first symptom expression, it varies widely from days to months. Asymptomatic carrier seeds pose greatest threat to plant quarantine. A seed lot may appear pristine, clean during visual inspection at customs, yet it may harbor latent bacterial or viral infections that manifest only weeks after planting in a farmer's field after import. When infected seeds are planted, the consequences unfold rapidly. Primary impacts include seed rot, pre-emergence damping off, post-emergence damping off, and surviving seedlings suffer from systemic stunting, vascular wilt, and reduced leaf area, leading to severe penalties that cannot be recovered later in the season. To summarize this section, the epidemiological foundations, seeds are highly efficient survival structures and primary inoculum sources. Though vertical transmission and latency, pathogens survive storage and travel long distances undetected. Controlling diseases at this initial stage is mathematically and economically the most powerful intervention available in plant pathology. The next section is that on pathogen biology. We begin with fungal pathogens, the largest group of seed-borne agents. Key genera include Fusarium causing seed rots and head blights, Alternaria causing leaf spots and black points in cereals, Escochita causing destructive blights in pulse stalks, particularly in chickpea, and Colototrichum that is responsible for anthracnose diseases across vegetables and legumes. It has wide range of its hosts. Major seed-borne pathogens are categorized into fungi, bacteria, and viruses or others. Key threats include Fusarium causing blight, Xanthomonas inducing bacterial blights, and seed-transmitted viruses or nematodes, all of which severely impact crop health, seed quality, and agricultural yield. This slide, as you can see, covers fungal seed-borne pathogens. Key genera like Alternaria and Fusarium affect cereals and vegetables by infecting the seed coat externally or embedding internally in the endosperm. They cause severe effects like seed rot, damping off, and reduced germination, surviving harsh conditions by specialized structures like sclerotia or dermatomycelia. Here we can see key fungal pathogens affecting beans, carrots, small grains, and maize in this slide. Please pay close attention to the unique diagnostic indicators for each, like orange spores or pinkish mycelium. Remember, Definitive identification requires combining visual inspections with laboratory incubation methods like blot or agar tests. As outlined in the current slide, we focus on 4 primary fungal seed-borne pathogens groups. Notice how Fusarium causes severe rot and blight, while Alternaria results in seed staining and quality losses. Furthermore, Ascochyta specifically damages pulse crops like chickpeas and lentils, whereas Polytotrichum, it induces destructive anthracnose across key vegetables and pulses. There are smut and bunt fungi like Stelago and Tilicia. They are specialized obligate seed-borne pathogens of immense economic importance. The teliospores replace healthy grain kernels with dark powdery spore masses, smelling of trimethylamine, that is fishy smell, stinking smut. And teliospores survive for years on the seed coat for inside storage facilities, germinating alongside the seed to invade the shoot effects epically. Bacterial pathogens account for 20% of global crop losses, and important species include Pseudomonas syringae, Xanthomonas campestris, and Clavivector michiganensis. Bacterial pathogens often persist inside seed coats or vascular tissues as latent infections, producing slimy vascular rouges and severe leaf blights once seedlings emerge under warm and humid conditions. This slide breaks down major bacterial pathogens by crop host. Key diagnostics include yellow ooze on beans, V-shaped leaf lesions on brassicas, greasy fruit spots on cucurbits, and distinctive bird-sized spots on tomatoes. Keep in mind, these bacteria are typically transmitted internally through the vascular systems or carried on seed surfaces. Seed-borne bacterial pathogens can trigger severe yield losses exceeding 20%, often hiding as latent infections. As shown in this slide, key drivers include Pseudomonas syringae, causing angular leaf spot and seed staining, Xanthomonas species behind bacterial blights and leaf spots, and Clavivector michiganensis that causes wilts and damaging systemic infections. How do bacteria maintain long-term associations with seeds? They rely on adherence mechanisms and biofilm formation. Bacteria secrete extracellular polymeric substances that form a protective matrix around cell clusters on the seed surface. This biofilm protects bacterial cells from desiccation, chemical seed treatments, and host immune responses, while type 3 secretion systems inject virulence proteins directly into the host cells upon germination. Viral transmission through seeds is a complex and highly regulated biological process. Viruses invade seeds either directly through parental vascular tissues via plasmodesmata or by infecting floral gametes, pollen, and ovules before fertilization. While not all plant viruses are seed transmitted, those that are, such as tobamovirus and potiviruses, they cause devastating early season systemic mosaic and stunting. The family Potiviridae that originates from potato virus Y is the largest and most economically destructive family of plant viruses. It encompasses over 200 recognized species of viruses. Seed transmission rates in potiviruses, it varies widely from 1% to 80%, depending on the specific host cultivar and viral strain. Viruses like bean common mosaic virus, BCMV, and lettuce mosaic virus are classic examples where seed health testing is vital to prevent total crop loss. In cereal crops, seed-transmitted viruses interact synergistically with insect vectors. For example, wheat streak mosaic virus is carried in wheat seed. Early emerging infected seedlings serve as immediate reservoirs for wheat and wheat kernel mites, which then spreads the virus across millions of acres. Biosecurity programs must monitor both seed health and vector populations to prevent widespread viral zoonotics. This slide highlights viroids and persistent viruses as specific pathogens. Viroids like phosphoroviroids in solanaceous seeds are highly stable naked RNA molecules without any protein code, subject to zero tolerance regulatory testing. Conversely, persistent viruses rely strictly on viral vertical transmission, causing asymptomatic infections that may offer stress tolerance or interact with acute viruses. This slide covers nematode and fumicides, which are less common seed-borne organisms. New and cryptic pathogens are continuously emerging. International seed trade passages. As climate zone shifts and exotic crop varieties are introduced, previously benign endophytes or localized pathogens mutate and adopt to new hosts, becoming pathogenic. Asymptomatic carrier seeds present a severe challenge as they show no visual symptoms. During shipping, it may cause severe blights and when planted in warm and novel environments. In seed pathology, we strictly distinguish between pathogenicity, the quality of the organism to cause diseases, and virulence. Virulence measures the quantitative degree or severity of that disease. Environmental stress during seed storage, such as high humidity or temperature fluctuations, can alter pathogen virulence, turning mild latent infections into highly aggressive strains upon germination. A single commercial seed lot can harbor multiple races or biotypes of a pathogen simultaneously. This genetic diversity poses a grave threat to plant breeding programs. When a seed lot containing diverse pathogen races is distributed across regions, it introduces rare virulent strains that overcome localized host crop resistance genes, and that may lead to sudden breakdown of disease resistance that is being harped upon. When infected seed lots are stored under subtropical humid conditions, seed surface fungi produce massive quantities of airborne spores. During seed conditioning, grading, and packaging, mechanical agitation releases these spores into the air, and that contaminates adjacent clean seed lots and creates occupational health hazards for seed factory personnel. Beyond direct crop losses, seed-borne fungi like Aspergillus, Fusarium, and Penicillium, they synthesize dangerous secondary metabolites that are known as aflatoxins. Trichothecenes and fumonicenes cause severe hepatotoxicity and cancer in humans and livestock. Regulatory frameworks enforce strict threshold limits for mycotoxins in grains, making seed health a major public health issue. Seeds are rarely infected by a single organism. They frequently carry complex multipathogen complexes. Co-infecting fungi and bacteria engage in competitive exclusion of or metabolic facilitation. For example, co-infection by Fusarium and Alternaria in cereal seeds can amplify total tissue rot and mycotoxin accumulation for beyond what either pathogen produces alone. So there is a synergistic effect between these two fungal pathogens. How long can pathogens survive in stored seeds? Fungal teliospores of smut species can remain viable inside stored grains for several decades. Can you believe? So, bacterial cells and plant viral RNA survives inside embryo tissues as long as the seed embryo itself remains viable. So, storage environments designed to extend seed longevity, cold temperatures and low moisture simultaneously extend the survival of the pathogen. Environmental conditions during international shipping dictate whether latent seed infections remain dormant or trigger severe seed decay. Cold storage, temperatures between 2 to 4 degrees Celsius, it preserves seed vigor and suppresses bacterial growth, but moisture condensation inside shipping containers by this cold temperature creates ideal microenvironments for fungal spore germination and decay in transit. Climate change is fundamentally altering plant pathogen epidemiology. Warmer winters, altered rainfall patterns, and extreme weather events allow tropical seed-borne pathogens to establish in traditionally temperate agricultural zones. Further, unpredictable weather stretches traditional seed production windows, increasing crop exposure to seed contamination pathogens for a longer time. Summarizing this section, fungi, bacteria, and viruses utilize sophisticated evolutionary adopted mechanisms from deep embryo colonization to biofilms and spore longevity to ensure survival in seeds. Controlling these diverse threats requires tailored specific diagnostic and management tools. Coming to the next section, mechanisms of seed infection. Seed systems and biosecurity, we formally distinguish between seed-borne and seed-associated pathogens. There are two terms. Seed-borne pathogens are integrated into seed tissues internally, for example, embryo or endosperm, and transmit systematically to the progeny. Seed-associated pathogens are external contaminants attached to the seed coat or debris, which can often be removed via physical cleaning or surface disinfectation. There are certain differences among seed contamination and seed infection. A comparison is presented in the present slide. And these are the external routes of contamination: environmental deposition, harvest and processing, contact, and insect-mediated transmission. The internal infection routes include floral pathway, vascular entry, direct penetration, and systemic route. A brief chart of all the routes is presented in the slide. Global seed circulation occurs across four distinct seed systems, each carrying unique biosecurity risk profiles. Formal commercial systems feature strict testing, certification, traceability. Farmer systems rely on local knowledge but lack diagnostic oversight. Informal relief seed aid prioritizes volume during emergencies, often bypassing testing. Finally, e-commerce represents an unregulated frontier where small packages bypass border restrictions. Formal seed certification operates under rigorous international frameworks established by International Seed Testing Association and national certification agencies. This multi-step process includes field inspections of seed crop stands, post-harvest laboratory seed health issues, purity testing, and germination scoring. Seed lot meeting these standards receive ISTA blue or orange certificates, granting international market access. Informal and farmer-saved seed systems supply over 80% of crops in developing nations. While essential for local food security and biodiversity preservation, these systems operate without diagnostic screening. Pathogens accumulate over successive generations, a phenomenon known as seed generation, resulting in declining crop yields over time. Can I know how much time I have? Pardon? Okay, should I conclude? Okay, so quickly coming to the management methods, apart from detection, let's come to the management of all these. Seed treatment. This final section is that integrated management of and future directions. Once seed health status is determined, appropriate treatments must be applied. Chemical treatments including systemic fungicides like metallurgyl and broad spectrum contact tracings. Physical treatments such as hot water therapy, 50 to 35 degrees Celsius of temperature water, or thermotherapy effectively eliminates internal bacterial and viral pathogens from seed coats without chemical residues, providing temperature collaboration is exact. There are biocontrol agents and seed microbiome. These are the tailored mixtures of complementary bacteria and fungi, and genetic resistance. Host plant resistance is one of the important measures of management. And coming to systems approach, integrated management, because no single intervention strategy is foolproof. Sustainable seed health requires holistic systems. This framework integrates clean foundation seed, genetic cultivar resistance, field sanitation, cultural rotation, target seed treatment, and climate controlled stage. Combining multiple defense layers ensures that if one barrier fails, secondary layers prevent widespread disease outbreak. These are some new methods. There are emerging technologies, and coming to research gaps and priorities, quantifying seed infection rates across crops and regions. Scientific community must focus on these 4 points: unraveling molecular triggers that activate latent pathogens during inhibition, optimizing synthetic microbial community, stability of seed rods, and 4th, modeling climate change impacts on seed-borne epidemiology. Concluding, seed-borne plant pathogens represent quite yet formidable threats to global food security, agriculture sustainability, and international trade as well. Protecting our agricultural future requires uniting cutting-edge molecular diagnostics, sustainable microbiome management, rigorous quarantine policy, and international cooperation. By securing the health of the seed, we secure the future of global food supplies. And thank you very much for your time, attention, and dedication to advancing seed health and plant biosecurity. I would like to extend my heartfelt thanks and gratitude to the conference organizers, Banaras Hindu University, and all collaborating institutions. I am now open to any questions, comments, or discussions. Thank you.
Roll in advertising India planetary exploratory programs and served as the principal investigator for the key scientist instrument of the Chandrayaan-1 Mars orbiter missions MOM
Chat
Chandrayaan-2,
you away with it.
Chandrayaan-3 and Aditya L1. His pioneering research in planetary atmosphere, solar system explorations and X-ray astronomy has earned international reason. Professor Bharadwaj has published 220 research papers and is a recipient of the prestigious Shanti Swarup Bhatnagar Prize, Infosys Prize and COSPAR Vikram Sarabhai Medal, among many others national and international honors. So sir, welcome you. I invited to you take over the session, sir. Sir.
Thank you. Can you hear me?
Sir. Yes, sir.
Can you hear me well? And you can see my slides?
Yes, sir.
Okay, good. So good morning. Namaskar to everyone, all the viewers and to the organizers. And thanks for organizing this conference and inviting me. So I'm going to take you to the moon. As you all know very well, Bharat Chandrama par pahunch chuka hai. And my talk is very preliminary in nature to tell you what is happening with respect to Indian exploration program of the moon and what we are planning to do in the future. So this talk is organized in terms of showing you some glimpses of what we have achieved and what we want to do in the future. So this slide, you must be seeing it very well, shows the Indian lunar exploration program starting from the Chandrayaan-1, Chandrayaan-2, Chandrayaan-3, the launch vehicle, the communication which we do using the big tele antennas and things like that. But this whole thing is based on how the space program in the country started under the leadership of Dr. Vikram Sarabhai. Let me see if the slides are moving or not. I hope the slides are moving. Can you see the change in the slides?
Yes, sir, it's moving.
Okay, great. Thank you. Perfect. So, Dr. Vikram Sarabhai, you started the Physical Research Laboratory from where I'm talking to you right now. And it started very soon, within few months of our independence in the year 1947. To be precise, on 11th of November 1947. Dr. Sarabhai established PRL, and that's why PRL is called as the cradle of space research in India. Because when you talk about the rocketry program, you talk about the satellite program, you talk about the ballooning program, application program, everything started under the leadership of Dr. Vikram Sarabhai. And here you are seeing this photograph with Dr. Sarabhai in the center. And this is former President of India, APJ Abdul Kalam, who was actually working with Dr. Sarabhai in PRL in Ahmedabad before he was sent to Tiruvananthapuram or Thumba to start the rocketry. So the first rocket launch, which actually took place from Thumba in the year 1963, on 21st of November, carried experiments developed in PRL. Okay? And the program which led to the development of ISRO, which actually happened in the year 1969. So PRL was formed in 1947 and ISRO came into existence in 1969. Okay? So that's why PRL is called as also the mother of ISRO. PRL currently has 4 major campuses. The main campus you are seeing in the top left. We have another campus in Ahmedabad, which is called Thaltej campus. And then we have 2 observatory campus, one in Mount Abu in Rajasthan, where we have big telescopes. It also has a 2.5 meter telescope, which is the second largest in India. And we have Udaipur Solar Observatory, which is in the middle of the lake, Fateh Sagar Lake, which is the largest solar telescope in India. Talking about the lunar exploration program, as you may be knowing very well that it all started in the year 2008 when we had the launch of the Chandrayaan-1. This was followed by almost a decade later on the Chandrayaan-2 mission, and then the most celebrated Chandrayaan-3 mission, where we landed for the first time in the southern polar regions. So I will give you brief glimpses of all these 3 as I move forward. So talking about the Chandrayaan-1 mission, we had the launch on 22nd of October 2008, arriving on moon on 8th of November, and carrying 11 experiments on board. It was a truly international with 10 different countries participating. And if you look at this satellite, I hope you may be seeing this picture. These are all experiments or payloads, we call. And they were meant to do science on the moon. And I was directly involved with the instrument called SARA, Sub-KV Atom Reflecting Analyzer, which are sitting on the top deck of this Chandrayaan-1. It also had a gray box, which you see in the top, absolutely top. And that was the one which was actually released from the mother spacecraft, the Chandrayaan-1 orbiter, and landed into the southern polar region, very close to the South Pole, making MIP the first man-made object to land into the South Pole of the moon, and putting the Indian flag, which happened on 14th of November 2008, okay, in evening around 20:36 our Indian time. So India had actually put its flag on the lunar South Pole on 14th of November 2008 itself. Okay. Now, talking about what science has come out, and I'm just putting one slide here just to give you glimpses, but the most important science result is discovery of water. Till that time, we were not knowing that water exists on the moon, but Chandrayaan-1 mission only told that water exists on the moon. It is there beneath the surface, on the surface, and above the surface. And of course, a lot of new science has come out, which has been published in more than 250 scientific publications talking about validating how moon got formed, which we call as lunar magma hypothesis, sputtering or scattering happening of the solar wind, new minerals were found, volcanic vents and lava tubes have been discovered, and how the solar wind interacts with the moon. Many of them actually made the cover pages of various international journals like Science, Geophysical Research Letter, which is published by American Geophysical Union, and of course our own current science journals. Let me talk about the Chandrayaan-2 mission, which was much more complex and complicated. Why? Because it not only had the orbiter like Chandrayaan-1, but it also had a lander to land on the lunar surface called Vikram lander, named behind Dr. Vikram Sarabhai, the father of Indian space program. And it also had a Pragyan rover, which is sitting inside the lander. Now, you all know very well that what happened on 7th of September 2019, early morning, when we attempted to land on the lunar surface by the Vikram lander, but we could not achieve success. But the orbiter, the Chandrayaan-2 orbiter, carrying 8 experiments on board, is still in orbit around the moon after close to 7 years and providing us excellent quality of data. And one of the instruments called OHRC, or Optical High Resolution Camera, is the one which decided the landing site for the Chandrayaan-3 mission. And this instrument is now being used by various space agencies, including NASA, ESA, JAXA, different countries around the world which are planning to land on the lunar surface, is utilizing the data from this optical high resolution camera of India on the Chandrayaan-2 orbiter. Why? Because it is providing us a resolution of almost like 1 feet from 100 kilometers on the lunar surface. And that's why everybody is asking for images from this mission for their choosing the landing site. We also had a dual frequency synthetic aperture radar, LNS band, and this provides you information beneath the surface to few meters. So not only you get the information above the surface or at the surface, but what is there beneath the surface. Also, we had many other instruments like CHASE-2, IIRS or CLASS, which is providing the distribution of elements on the lunar surface, distribution of water on the lunar surface, elements, or distribution of argon, for example, across very close to the lunar surface. Now, let me quickly go to Chandrayaan-3 and tell you about how we went about it, because this is, of course, a very important mission from India, where we achieved the landing using the Vikram lander, and the Pragyan rover came out from the lander and moved on to the lunar surface. So the launch of this happened on 14th of July 2023. So it's about 3 years back at middle of the day. using the GSLV Mark III, which is the heaviest lift-off vehicle. So here you can see the lander, the Vikram lander, sitting over the top of the propulsion module. Why we went with the propulsion module? Because Chandrayaan 2 orbiter with 8 experiments are still in orbit. So we don't want an orbiter. We want only a vehicle, the module, to carry the Vikram lander in orbit around the moon so that we can do the landing. So this is in the clean room where you are seeing this Chandrayaan 3 lander, Vikram lander, sitting on the top of the propulsion module. And this Chandrayaan 3 lander had 3 experiments, in addition to, of course, the NASA's lunar retroreflector. And we had 2 experiments on the rover. And PRL has an instrument called APXS, Alpha Particle Access Instrument, to determine the elemental composition of the lunar surface on the rover. But also on the lander, we had an instrument called Chaste, which is like a thermometer going inside the lunar surface up to a depth of 10 centimeter to look at the temperature distributions in the top 10 centimeter. We also have a seismometer, ILSA, and also a Langmuir probe, which is providing us the plasma distribution very close to the lunar surface at a height of about 2 meters. So these were the instruments on the rover as well as on the lander. But we also have a small instrument called SHAPE, which was there on the propulsion module, because propulsion module went on into an orbit around the moon for more than 2 years. And this also has been used to understand a lot of science of how the Earth will look like from the moon, so that we can use that to understand how exoplanets or planets outside our own solar system will be. Now, talking about the launch, I hope you can see this slide very well, where I'm showing you the Sriharikota, the launch pad of India, and you are seeing this GSLV Mark III, which we call as LMV3, with the Chandrayaan 3 lander along with the propulsion module sitting on the top. So you see here the launch vehicle. In the top portion, you see the heat shields, which is there, and inside that is the Vikram lander sitting there. And let me see if I can show you the launch, if it is possible. Okay, so you are able to see the launch, which happened on 14th of July 2023 in the middle of the day. And just confirm to me, I am going to show you the real launch of how it takes place. Please confirm if you are able to see it, okay? And also listen to the voice.
3, 2, 1, 0.
Are you able to see it?
Yes, sir.
And listen to the voice.
Here we have a majestic liftoff of LVM-3 M4 rocket.
So this was the launch of the Chandrayaan 3. This is how the rocket launch takes place. And LVM-3
रॉकेट आसमान को चीरते हुए अपनी भयानक दहाड़ से चारों ओर कंपन उत्पन्न कर रहा है.
Are you able to listen to the sound?
और यही दहाड़ है जो वैज्ञानिकों को रोमांचित करती है.
Okay, good.
LVM-3 के उत्थापन के साथ ही हमारे अंतरिक्ष यान चंद्रयान 3 की चंद्रमा की यात्रा प्रारंभ हो चुकी है.
As the rocket is soaring through the clear skies, every second moving closer to the accomplishment of the most important milestone in its mission to moon.
Okay, so this was the launch. And after the launch, we went to an orbit around the Earth, then went into a lunar transfer trajectory, then went into a lunar orbit. And finally, we went into a 100-kilometer circular polar orbit, and then came down to 100 by 30. So the landing actually started from 30 kilometers down to the lunar surface. And you know that it happened on 23rd of August 2023, which we now celebrate as the National Space Day. So I'm now going to show you, this was the scene in the mission control. I was also present there watching very keenly. You can see the current chairman, the former chairman, all center directors of ISRO's units, centers sitting there. So now I will show you the last part of this mission profile, where we are waiting eagerly to see how the launch takes place. We are quite confident, but then, of course, this is always a challenge. So I'm going to show you this as a live. Okay.
Sensors that are updating at this point are providing confirmation of the safety of the landing site.
As expected,
Oops.
the retargeting is going on, and this is a very good signature for the lander. अभी आप ये देख सकते हैं कि लैंडर की ऊंचाई लगभग 65 मीटर के आसपास है.
So this is about just 63 meters above the surface. We started from 30 kilometers, right? So you have come very close to the lunar surface and about to land. Prime Minister of India was there in South Africa during that time. So he was watching live from there. So we are coming very close to the lunar surface, just 50 meters above.
और हम लगभग 50 मीटर से भी कम आ चुके हैं.
And in the middle, you must be seeing the lunar surface, how the lander was actually seeing the lunar
You can
surface.
see the Honorable Prime Minister, Shri Narendra Modi ji, who is here to encourage us, and he is critically looking at the visuals.
So we are very close to the surface now, and we have landed.
स्क्रीन पे देख सकते हैं कि हम अब लैंडर मॉड्यूल
People are applauding. Let us all wait to hear from the Secretary Department of Space and Chairman ISRO, Sri S. Somnath.
So this moment always give me the goosebumps. Why? Because I was also there in the mission control, watching it live, and we were absolutely jubilant. So everybody was feeling it over the moon. So I also felt over the moon, and I took the selfie with the Vikram lander and Pragyan rover. But then, how was the world actually responding on this? Okay, just listen to this small clippings from different news channels around the world.
India has done it. It has become now the fourth country to land a spacecraft on the moon after the US and China and the former Soviet Union. Just moments ago, we're talking to
India has made space history, becoming the first nation to soft land a spacecraft on the moon's south pole. India has become the first nation to successfully land a spacecraft near the south pole of the moon. Country's PM Narendra Modi has praised the achievement, saying that mission success belongs to all of humanity.
The spacecraft Chandrayaan-3 is attempting to be one of the first to land on the moon's south pole.
India is the first country in the world to land a spacecraft near the moon's south pole, a moment of history for the country. It was at tense final 6 minutes as it descended onto the lunar surface. Let's show you the moment. First of all, let's take
This agency is celebrating the successful moon landing of its Chandrayaan-3 spacecraft.
Okay, now India's lunar probe Chandrayaan-3 has landed on the moon's south pole. India is the first country to touch down on this part of the moon, which is believed to hold pockets.
We begin with some historic news. India has become the first country in the world to successfully land a craft near the moon's south pole. Prime Minister Narendra Modi was watching the mission from South Africa and congratulated the team back home. Let's take a closer look at what.
So this is how, the whole world was celebrating the success of the Chandrayaan-3 landing, which made India the first country in the world to land into the south pole of the moon. Now, after the landing, we are all not very jubilant because we had to have the next step, which is taking out the Pragyan rover after the landing. So here you see in the mission controls in the clean room in Bangalore, we have done several tests. Will come out and roll down to the lunar surface, but now we have to do it on the lunar surface, right? And this was the first time we are doing it. So we are all very excited, but also very nervous to see how things will unfold. So I will now show you how it actually happened that the rover came out on the lunar surface. So this is the rover. It is getting released. This is happening on the lunar surface, and we are all sitting in the mission control watching it live. You can see the shadow of the rover. You can see the Indian flag and the Indian symbol, emblem. So the rover started rolling down, and we could see the, the tracks formed by the lunar rover, the Pragyan, or the lunar surface. And then the rover took the selfie of the lander. And this is a very unique photograph of Indian Chandrayaan 2 Vikram lander sitting on the lunar surface at the Shiv Shakti point. After this, we deployed different instruments on the lander to do the science. So you see here in the bottom, the Chaste instrument, which has gone down to 10 centimeter into lunar surface, ILSA, which is providing the seismicity, and the Ramba LP, which is basically to sense the plasma distribution around the landing site. And we all know that this landing site is now known as Shiv Shakti, and it has been now confirmed by International Astronomical Union that this landing site of Indian Chandrayaan 3 will be called as Shiv Shakti. Now the rover which has come out has moved onto the lunar surface. So this is how you are seeing the actual track of this rover. We started moving to one side. We found a big crater in the front. We moved back. We started going to the east. But then there were a lot of planetary scientists, planetary geologists, mission controllers. And this geologist said, no, we should go to the west side because there's a new crater which is sitting here on the left. And therefore, we started moving to that direction. So this whole track which you're seeing is almost like 103.5 meters, which was covered in about 10 days of the Pragyan rover moving onto the lunar surface. And this payloads which has gone on the lander as well as rover has provided us high quality of science data. More than 50 scientific publications in very reputed international journals has come out. There's just a glimpse of what science has come out from the instrument. For example, the top left, you are seeing the temperature as we go down. So you are seeing from the top to almost like when you go to a depth of about 10 centimeter, you are able to see a temperature distribution of about 50 degree Kelvin, which in turn means that the temperature from the top or the heat from the top is not going down. We saw a lot of seismic activities. So you are seeing ILSA results, you are seeing the LIPS result providing a spectrum, or the Ramba LP providing what kind of plasma densities are there very close to the lunar surface. Though the moon is not having much atmosphere, but still there is a plasma very close to lunar surface of a densities of about 1,000 particles per cc. And APXS, which is an instrument developed in PRL, has provided us various elements present around the landing site. So you can see here the rover, and this instrument was right sitting in the front, and this plate which you are seeing used to get deployed as we want to take the measurements. So when the rover was stable, this plate will be deployed down. It will take measurements for a couple of hours, and then the plate will come back and the rover will move. So we made measurements at almost 30 different locations on this track which you have seen, which I have shown you, providing us elemental composition around the landing site. And this results have now been published last year in the journal Nature, where we have showed that what is the actual distribution of elements at the lunar site, landing site of the Chandrayaan 3, and has also showed that there is a larger amount of magnesium, which is quite a surprise, telling that probably material from the inside, probably from the South Pole African Basin, has come at this landing site, showing a higher distribution of magnesium. So I'm not going to go into details of the science because it is more like a popular talk. But what we want to do next, very quickly in next 2 minutes. Now the future is for the Chandrayaan 4 mission, where we are talking about the sample return. So that means we have orbited, we have landed, we have moved onto lunar surface. Now we want to bring back sample to the Earth from the moon. So this is a mission called Chandrayaan 4, which is going to be a very complex mission because we are going to have 2 launch vehicles carrying different modules, and these modules will go into an orbit around the Earth, then they will go into an orbit around the moon. Some part will be in orbit around the moon, some part will land on the lunar surface, then collect the sample. A small ascent module will come back to the orbiting module around the moon, and then that will bring it back to the Earth's orbit, and then finally onto the Earth's surface. So this is a very, very, very complex mission, but this is also technologically challenging because there's been a lot of docking and undocking happening in the orbit around the Earth and around the moon. And we want to try this mission as a Chandrayaan-4 mission. We are quite confident that we'll be able to achieve this, and this will bring back the samples from the lunar surface. The next mission is the Chandrayaan-5 mission, which is also a ISRO-JAXA or India-Japan joint mission, where we actually want to land into the South Pole of the moon. You see here the arrow along with the latitude, longitude given, minus 89.44. So minus 90 means exactly poles, right? But here we want to land into a very narrow strip, which is eliminated by sunlight, because we need sunlight to produce power on the spacecraft, on the lander, and the rover, which has to go inside into this permanently shadowed regions to provide us absolutely new data, because nobody has gone inside this permanently shadowed regions where sunlight never reaches, because in the poles, if you go, and the moon is almost vertical, right? It is not tilted like Earth, 23.5 degrees. So in the case of moon, if you have craters in the polar regions, they never see the sunlight. And therefore, these regions are called permanently shadowed regions, where the temperatures could be as low as almost like 50 Kelvin. And the idea is to go inside and to see what is there. So therefore, ISRO is providing the lander, the JAXA is providing the rover and the launcher, and we have instruments on the rover, both from India and from Japan. And this is going to be a joint operation mission by India and Japan, where there are science payloads coming from other agencies like NASA and European Space Agencies. So this is also a very, very complex and very important mission for not only from the science perspective, but from humanity perspective, because we'll be landing for the first time very close to the South Pole. We are also talking about future missions like Venus orbiter mission and Mars lander missions, where we are going to orbit Venus for the first time to do a lot of new science, which has not been achieved by any mission previously, and also talking about landing onto lunar to the Martian surface. So these are all missions in the future, which you must have also heard about through media as well as from ISRO. And so there's a lot of bright future for youngsters who are interested to join the exploration program of the country, where we have now a roadmap going to go to the moon, not just for the Chandrayaan-4 and 5. We are talking about Chandrayaan-6, 7, and 8. We are also talking about the Bharatiya Antariksh Station and Indian landing onto the lunar surface by 2040. So the future is quite bright, and I thank you all for listening to this talk very patiently, and invite you to join the space ecosystem and the space program of the country. Thank you. Thank you very much.
Thank you so much, Professor Anil. On behalf of the organizing committee, I extend our heartfelt gratitude to you for delivering such an enlightening and inspiring session on Chandrayaan. Anybody has any queries or question want to ask to sir anything? All the participants or panel?
Good morning, sir. Anand Srivastav, and I would like to ask one question on Chandrayaan-3.
Yeah, go ahead.
Sir, is there any plasma experiment is deployed in the Chandrayaan-3 mission?
There was an instrument called Ramba LP, which I was just mentioning, which was there on the lander. And it was sitting at the top deck of the lander to measure the plasma, or basically the electron densities very close to the lunar surface at a height of about 2 meters. And what we have seen is that densities of about few thousand particles per cc is present there. So this is a very unique and the first observation of its kind in the whole world.
So what type of temperature we will have there?
Sir, you are not audible. Professor Anil?
Okay, can you hear me now?
Yes,
Yes,
sir.
please.
Okay. So, are you able to hear me the last answer?
No, sir. No, sir. Kindly repeat.
Okay. So, the plasma distribution at the landing site has come out from the Chandrayaan 3 lander instrument called Ramba LP, which provides us information telling that plasma of about few thousand particles per cc is present at a height of about 2 meters from the lunar surface.
Okay.
About the temperature distribution, it has come out from the instrument called Chaste, which was there on the lander, which has gone inside the lunar surface to a depth of 10 centimeter. And almost like every 1 centimeter, we have the temperature measurements. And that is one which has told that the heat is not propagating down because the lunar regolith or the lunar soil is highly non-conductive. So there are publications which has come out. You can simply search in Google Chandrayaan 3 and you will see all the science results.
Thank you. Thank you, sir.
Okay, thank you so much because I had to go for another meeting.
Yes, yes. Thank
I'll
you
take
so
leave
much,
from
sir.
you. Okay, thank you. Take care. Bye-bye.
Okay, thank you, sir. Now we will move forward to our next session. Good morning, all the distinguished guests, respected chair and co-chair, presenters and participants. It is my pleasure to welcome you all to the presentation session 9, day 2 of the 5th Global Conference on Emerging Trends in Research and Development, ETRD 2026, being organized in hybrid mode. I am Dr. Rekha Sharma, and today I am having the privilege of serving as a moderator for today's virtual technical session 9. We are honored to have with us Dr. Shiva Soni as chair and Dr. Shirin Sahu as co-chair. Now I would like to introduce our chair and co-chair. Dr. Shiva Soni is an associate professor in the Department of Physics at Purnima University, Jaipur, Rajasthan, India. She earned her PhD in physics with a specialization in space physics from Avdesh Pratap Singh University in 2015. With more than 15 years of teaching and research experience, she has made significant contributions to the fields of space physics, astrophysics and astronomy, solar astronomy and plasma physics. Dr. Soni has published over 60 research articles and has presented her work at numerous national and international conferences and seminars. She has actively participated in faculty development programs, workshops, and short-term courses, and has contributed to academic events as a session chair, organizing committee member, keynote speaker, and editorial board member of reputed journals. Her teaching expertise include quantum mechanics, nuclear and particle physics, atomic and molecular physics, laser, and spectroscopy. She received the Young Scientist Award 2022 and the Distinguished Research Award ETRD 2023 from the International Journal of Innovative Research and Growth. Our co-chair, Dr. Shirin Sahu, is an assistant professor in the Department of Zoology at Government Vivekanand PG College, Maihar, Madhya Pradesh. He has published 20 research papers in reputed national and international journals and has actively participated in several conferences, seminars, and workshops. Dr. Sahu is actively engaged in teaching, research, and mentoring students, contributing to academic excellence and interdisciplinary research. On behalf of the organizing committee, I also extend a very warm welcome to all our presenters and attendees joining us from different institutions and locations. Now we will move forward. Today's session will feature our poster presentations covering diverse and emerging areas of science, technology, and interdisciplinary research. Now keep your microphones muted unless invited to speak. Turn on your cameras during presentation, and if possible, adhere to the allotted time, and reserve the final minute for your questions and comments from the chair and co-chair. Now I would like to invite our first poster presenter, Dipanshu. Are you here with us? Okay, next is Aman. Aryan Sharma.
Yes, ma'am. I am on a Aryan Sharma team, but he was not able to come today
Okay.
due to some reasons. So I am going to present.
Okay, Gaurav Rathi.
Yes, ma'am.
Okay, you will have to wait. Just two more participants are before your turn. Saurabh Chaturvedi.
Okay.
Ganeshwari Singh, Dipra Arora. I guess they yet to join. Okay, Gaurav, you present your poster.
Okay, ma'am. Ma'am, my screen is visible.
No.
Now,
Not yet. I still can't see anything.
Maybe it was some kind of problem.
Now it is visible, but it is just a gray slide. Nothing else is showing. It's not showing anything. It's just a gray blank slide.
From my side, it was like,
I try to share it again.
just a second. I think this one. No, let's see. Okay, is it visible now?
No, Gaurav, it's still not visible. Are you sharing? Yeah, now it is visible.
Yes, ma'am, I'm sharing.
Yeah, it's the digital number system.
Yes, ma'am, it is digital
Yes,
number
yes,
system, but the way proper name was optimize healthcare workflow with Salesforce CRM. So,
your just
good
slide
morning,
is visible
everyone.
in which it is.
I am Gaurav Bagdi.
Shiva ma'am?
Yes, ma'am.
Ma'am,
Can I start, ma'am?
just wait for a second. ID ma'am 267185.
Okay.
Okay, now Gaurav, you can start.
Ma'am, my ID was 267143.
It's Gaurav Rathi,
Okay,
right?
ma'am. So, Gaurav Bagdi.
Gaurav Bauri.
Ma'am, I'm
Just
on Aryan summer team.
okay,
This
Aryan.
is
You
on
are
group project.
presenting on behalf of Aryan.
Yes, ma'am. Due to some reason, he can't able to present
Okay.
right now.
Okay, so 267143.
Yes, ma'am.
Okay, fine.
So, good morning, everyone. I am Gaurav Bagdi, and today I am presenting on our project, our optimization healthcare workflow with Salesforce. Presentation will explain the current problem in healthcare management and how Salesforce can help improve it. So, in hidden cost fragmented optimization, in many healthcare organizations, systems are discontinued because of patient record, scheduling, inventory, billing, and etc. Together smoothly, this create delay, increasing manual work, and affect patient care. So, as we can see, traditional methods were not properly used it was very lengthy. And in a Salesforce-based system, existing systems have limited automation, poor conditions, but Salesforce make it workflow automatically, inventory management, and centralized optimization. Salesforce make it much easier to use by anyone who was used in the system and in their fields. Salesforce also unified cloud environment. How we can see in our Salesforce beginning different healthcare care functions in one unified cloud environment. Inventory of work, separate departments like patient records, scheduling, administrator inventory. Now, this was our system architecture in which receptionist, doctor, pharmacist, or administrator, also patient, can be access this program and use their respective areas like in laboratory, receptionist area, or medical laboratories. This was fully automated and make workflow easier and make all the process interconnected. So in this diagram, this page, here hospital functions are matched with Salesforce technology. Example, appointment management, doctor requirement are established in the percentage. It was calculated by a Salesforce cloud system, which can be used with Salesforce flow and Apex class, also with trigger and validation rules. This can make work more fluently and give automation suggestion for the user and the applicant. Here, hospital functions are much with Salesforce technology. For example, appointment, doctor requirement, admission, billing, etc. Salesforce trigger flow are much helpful in this program to make it automated and connect with everyone. It also generate alert when inventory or some kind of mishap is occurred. This function on Salesforce make hospital system are much better for using for everyone. This slide focuses on appointment, scheduling, and admission handling. Salesforce Flow Trigger help providing duplicate booking and automatically assign beds based on emergency priorities. Alert-based inventory management is very unique in this system. If medical stock goes below a set limit, the system sends an alert to administrator to help avoid storage shortage and ensure the improved medicines are available here. It also give alert when some kind of emergency are appeared in the customer health and on many more. This slide explains how billing become easier with automation. Charge for consultancy, medicine, and lab service are counseling automation, which reduce manual work and provide prevent billing error. The billing process automate. Yes, ma'am.
Your slide is not moving, and also try to conclude in next 2 minutes because we have time constraint.
Okay. So I'm now conclude. Salesforce creates strong function for future healthcare. It supports central CRM workflow automation, predictive analysis, and better health hospital management. Thank you. And
Are you done, Gaurav?
yes, ma'am.
Uh,
Any
okay, I would like to ask now panel or the participants if they want to ask anything. Yes, anyone from the panel or the participants? Okay, thank you so much, Gaurav, for the presentation.
Thank you, ma'am.
Okay, next I would like to repeat again the names who are absent. If you have joined the meeting. Dipanshu, Anam, Saurabh Chaturvedi, Gyaneshwari Singh, Dipra Arora, and Gaurav Rathi.
Yes, ma'am, I'm here.
Gaurav Rathi?
Yes, ma'am.
Yes. Start your presentation. Ma'am, ID is 267185.
Yes, ma'am. 185.
Yes, ma'am. 185.
Okay. Okay, start, Gaurav.
Okay, now I'm sharing this.
Gaurav, you have 5 minutes. Just try to conclude in 5 minutes.
Okay, ma'am.
Okay, you can now start.
Ma'am, I'm unable to share screen at the time.
Try it one more time. Yes, your presentation is visible. Okay.
My presentation is visible or not, ma'am?
Yes, it is visible.
Okay, thank you. Very good morning to all of you. My name is Gaurav Rathi. I am a research scholar in Bhagwant University, Ajmer, Rajasthan. My research guide is Dr. Rekha Ashrani. My topic is a regioselective synthesis of novel thiazole derivative and spectral characterization with potential medical application. Now introduction. This is thiazole. Thiazole is a 5-membered heterocyclic organic compound bearing both nitrogen and sulfur at position 1 and 3. Thiazole is an important intermediate in chemical synthesis, drugs, dyes, and in all other bioactivities. Thiazole is using thiazole derivative is used as a anti-cancer, and many other disease. Thiazole derivative used, and our main objective is to synthesize thiazole derivative, whether the presence of 3 group in the same molecule and increased its antioxidant effect. To synthesize thiazole derivative, characterize them by the proton NMR LCMS technique and purified by HPLC or column chromatography to evaluate thiazole antioxidant and antimicrobial activities. And this is our main scheme. Firstly, we started with acid, and then in step 1 we made acetamide by using thionyl chloride and ammonium hydroxide. We successfully got intermediate 2. Then we made thiamide by using the Lawson's reagent. Intermediate 3 is a purified by column chromatography. Now in step 3, we made the thiazole ring by using ethyl chloroacetoacetate as a reagent in ethanol, and we made intermediate 4. And this is our, you can see this is our thiazole ring. Then we hydrolyzed it and formed acid, and again we doing amidation reaction on intermediate 5 with different amines and synthesized compound 1 and other different compounds. Intermediate 4 is also used for next step by using reduction reaction followed by chlorination by using thionyl chloride, and made a common intermediate which is further used for the next final compounds. Here R is different benzyl group, R1 and R2, which is not disclosed at that time. So the structure and functional group of synthesized compounds were confirmed by NMR spectroscopy and LC-MS. HRMS analysis was performed to verify molecular weight, and the melting point of synthesized compounds were checked for purity and stability determination. So thiazole moieties have occupied a pivotal position in modern organic and medicinal chemistry due to its broad spectrum and pharmacological or medicinal activities such as antimicrobial, anti-cancer, antioxidant. The presence of thiazole ring in many drugs such as penicillin, thiazofurin, meloxicam, and nizatidine motivates the chemist to design new thiazole scaffolds for biological activities is awaited about this compound. This is in progress. So that's all. Thank you.
Okay. Thank you so much, Gaurav, for the presentation. Any queries from the panel or the participants?
Gaurav, actually,
yes, sir.
Can you define thiozole's actual function? What's their function in cell division? How it?
Sorry, sir, you are not clearly audible.
Actually, I'm saying to you, the actual function of thiazole in the cancerous cell. What their physical activity? How it prevent their cell division or their replication, transcription, translation?
Sorry, sir, I'm not getting your question.
Thiazole.
Hello? Yes.
Am I audible? Hello.
Sir, very little bit. I'm not clearly hearing you.
Sir, your voice is echoing.
Actually, I'm saying to you, what is the actual function of thiazole? How to prevent cancerous cell? Thiazole, how to prevent cancerous cell?
Madam, if you listen the question, then can you translate?
Shiva ma'am, could you please explain what sir is asking?
I'm asking about anti-cancer activity.
Ma'am, I'm not able to hear clearly, ma'am.
Okay. Yes, sir, please ask again.
What is the actual function of thiazole? In
He is asking actual
The thiazole derivative is used in many drugs we synthesized earlier, and many drugs are patented. So our main motive is to do research on thiazole derivative because we got different drugs made earlier by using thiazole, and you can see such as penicillin, Mexicam. So our main aim is to make a thiazole derivative drug and see the biological activity in different compound.
Okay, thank you.
Okay, thank you so much, sir, and thank you so much, Gaurav. Now we have come to the end of our presentation session 9. I would like to express my sincere gratitude to all the presenters for sharing their valuable research work and insightful presentations. I also extend my heartfelt thanks to our respected chair, Dr. Shiva Soni, and co-chair, Dr. Shireen Sahu. I now request our respected chair kindly provide his concluding remarks or observation on today's presentation.
तुम्हारा तो है ही नहीं इस वाले में।
सेशन 11 है आपका सेशन।
सेशन 11 तो ज्वाइन ही नहीं करा रहा।
पूछ लो ना इसे।
Gaurav, please mute yourself. I would now request to Dr. Shireen Sahu to kindly provide his concluding remarks.
Hello. Am I audible?
Yes, sir.
Actually,
Okay, thank you everyone for your enthusiastic participation and cooperation. We wish you all your continuous success in your academic and research endeavors. Have a pleasant day ahead. Thank you everyone. Shiva ma'am, next session जो है plenary speaker.
Okay, and this section will be ended. Now we are going to the physical sections. So online section getting be closed right now. We are going to for physical sections right now.
Okay, sir. Thank you. Okay.
Dr. Dipender Haman, sir, roundly presented here. Dr. Dipender Haman. Yeah. Now I requested to Dr. Heminder sir, please present here. Now the
Good morning, everyone. It is privilege for me to be here today as a special guest at an international conference on emerging trends in research and development 2026, ETRD 2026. I sincerely thank the organizer, Vivekanand PG College, Maihar, Madhya Pradesh, for their kind invitation and warm hospitality. Thank you. Now, I am going to present my research, relativistic tight binding models for hexagonal lattice, application to graphene and extension to magnetic field effect. So as you know, in recent year, graphene has got a much more attention from the researcher because of its unique electrical, thermal and mechanical properties that makes it a promising material for future electronic devices. However, the detailed investigation of its properties has not been revealed yet for optimizing its electronic devices. So as you know, graphene is, due to this property, this is the unique material, one atom carbon atom, extraordinary electric honeycomb lattice, zero band gap, near Dirac point, strong and flexible material, high melting and boiling point, mass-less Dirac fermion, optically transparent material, high carrier mobility, large specific surface area, unique quantum phenomena. So up to now, the isolation of beginning of the research start in graphene is 2004. After a few years, 6 years, this material got Nobel Prize. In 2000, these are the graphene research up to 2026. 2013, graphene emerged as a promising material for flexible and transparent electronic devices, and 2018, the discovery of the superconductivity in twisted bilayer graphene by magic twist angles. So in 2021, graphene advances the quantum technologies through the development of high performance quantum devices. In 2024, it enables faster, energy efficient electronic and optoelectronic devices. So I think graphene is a driving next generation of AI hardware and quantum technologies. So application of graphene is because of its unique properties. It is high speed, we can make high speed electronic devices and we can make gas and biosensors and high energy density. Due to high energy density and fast moving charge, we can make nano devices. We can also make the super capacitor flexible and transparent display. We can make solar cell efficient and transparent by efficient and transparent conducting electrodes. We can also use in photonic and optoelectronic quantum devices and computing. We can use in spin-based devices too. So it is the composite material like stronger, lighter and more durable material. It has also the biomedical application too. So how I motivated is we develop a novel models, theoretical models that is used in the two-dimensional material, and we applied this method for the silicon, which is relativistic. We developed the relativistic tight binding model, which is applied for two-dimensional material. Now we are trying to apply this method to graphene. We already applied this method to graphene at zero magnetic field. Now we are planning to try this method in graphene, in the presence of graphene, to study the different electrical and quantum transport phenomena. So there are some method, there are some tight binding method, non-relativistic and relativistic tight binding method already present. In some method, non-relativistic method, tight binding method applied to some material with zero magnetic field and in the presence of magnetic field. And there are also some relativistic tight binding method in the presence of zero magnetic field. But there are no such method which has both spin orbit or in spin orbit interaction and magnetic field. So we thought to develop a new method which contain both relativistic effect or spin orbit interaction and magnetic field. So we developed a method which contain both effects simultaneously. So this is the non-relativistic tight binding method to find the electronic structure of material by using the Schrödinger's equation. SI is equals to E psi, SI is equals to E psi, where the Hamiltonian of the atom is given by this equation. Now the Schrödinger equation for an electron that moves in a crystal with periodic potential is given by last equation in our slide. So after rigorous mathematical calculation, we get the Hamiltonian of the matrix by this equation, where T is the Hoping integer. This means coupling strength between the lattice sides. So why need the relativistic model? Because since the tight binding approximation method solve the non-relativistic Schrödinger's equation, the relativistic effect like spin orbit interaction and magnetic field are not taken into consideration. So in order to have more accurate coherent tight binding models incorporating both magnetic field and relativistic effect, so we need to start from the relativistic Schrödinger's equation called the Dirac equation. So we developed the Dirac Hamiltonian by using the Dirac equation. The relativistic effect like spin orbit interaction will inherently included in our calculation. So obtained electronic structure will be relativistic electronic structure. So this is one of the novel method which calculate the electronic structure of two-dimensional material. So in relativistic tight binding approximation, we use the Schrödinger's equation E psi equals to E psi, where the Hamiltonian is the Dirac Hamiltonian is given by C alpha dot P beta MC square. And this is the potential. So where alpha and beta are the 4x4 matrices, which helps this Hamiltonian to make the relativistic one. So using Bloch's theorem and writing the atomic orbital, we get the crystal wave function is given last of this equation. So I do not go mathematical part through, and after the rigorous calculation, we get Hamiltonian of this matrix. So we apply this method, Hamiltonian in two conditions, at origin and at somewhere else. The atom first, initially atom is at origin and another is atom at somewhere. So this is the final equation. After much more calculation, we get final equation, simultaneous equation. This is our, we apply tight binding method to graphene, and we consider unit cell. Unit cell consider two atoms at a position Rn and Rn plus di from our origin. So this is the lattice site A. We solve this is the mathematical calculation. We after constructing the Hamiltonian and overlap matrix of the nearest neighbor interaction, we get the final Hamiltonian and overlap matrix. We get the final equation. Last, it is given. Si is Si. So finally, we calculate the electronic structure. Before calculating the electronic structure, we need the high symmetry point in K space. So we choose the high symmetry point in K space with gamma, m, and k with coordinates is given here. So in our calculation, we make the counter k, gamma, m, k as chosen. So this is the energy band calculation in our method. At the K point, the conduction band and valence band meets with each other, zero band. So graphene is also called the zero band semiconductor. So this figure just show the at Dirac point we get the two Dirac cones. So why magnetic field is, because the magnetic field strongly modifies the graphene's property. So why magnetic field is needed? Because the magnetic field strongly modifies graphene's electronic properties. It helps to quantize the electron motion, which is called the Landau level. It gives the very precise electrical resistance, standard used in quantum metrology. It helps to study the fundamental quantum physics like Dirac, fermion, and Berry phase. It splits the electronic spin into energy level, controls the electron transport by tuning magnetic field, controls the electron spin for spintronic application and used for quantum electronic devices, magnetic sensor, and spintronic research. So this is the, we currently extend before we use our method in graphene in the presence of, in the absence of magnetic field. Now we are planning to extend our method in the presence of magnetic field. Left side figure just so in the absence of magnetic field, this is the graphene energy band, and after magnetic field the energy band looks like in discrete. So major discovery up to now in graphene is zero electrical resistance as magnetic twist angle. Quantum Hall effect is observed at room temperature. Electrons move with exceptionally high speed, conductive and nearly transparent for optoelectronic, enables battery charging speed and performance, enables high speed terahertz communication and imaging, opens new, I think it opens new possibility for future quantum technology. So this is the how the graphene is expected to become one of the most important advanced material for future technology. This graph shows that. So why graphene is most important? Because we compare the property of silicon and graphene. So graphene is the silicon has band gap 1.12 electron volt, but graphene has zero band gap, no natural band gap. So it is also called the zero band gap semiconductor. Silicon has the moderate mobility, it has very high, and speed is also electron speed is high, and it's ultra high. It is built on, it is 2 times stronger than steel, moderate and extremely high thermal conductivity, which is rigid silicon, but it is graphene has flexible and bendable material. So with this much more application, it has some also limitation too. So it limits switching application because of the zero band gap. Large scale production remains much more expensive. I think uniform and defect free growth is very difficult. Metal contact reduces the device performance, we hope so. So graphene devices are difficult to integrate with existing silicon based CMOS complementary metal oxide semiconductor technology. I think it needs new technology. So in conclusion, we developed the relative basic tight binding method which contain both magnetic field and spin orbit coupling effect and applied to silicon with zero magnetic field case. Now we are planning to this method to in magnetic field. So I think graphene remains one of the most remarkable promising material for future technologies. So understanding these magnetic field may contribute the quantum, electronic and spintronic devices in future. Thank you.
Anyone can ask any question? Thank you for such a giving a lovely speech. Now I invited for next plenary speaker, Dr. Shankar Prasad Chimoria. Now I requested to Dr. Shankar Prasad Chimoria, sir, to be delivered the lecture here.
Okay, good afternoon. First of all, I'd like to thank you for organization committee. This is my second visit in Maihar, and this special thanks goes to the organizing committee for their nice hospitality and accommodation. I'm going to deliver my talk on the topics Josephson junctions, which is ultimate devices for AI enhancement. This is actually why we need this Josephson junction. Nowadays, the present situation, we are on the era of artificial intelligence. That's the reasons we should change the present network systems with based on the CMOS. That's the reasons we should be motivated to modify the present network systems. Then artificial intelligence is revolutionize the health sector, education, transportation, defense, and scientific discovery. However, there is a hidden problem. The problem are the present CMOS systems networking is the problems, and artificial intelligence demands the high computation, high data processing, because the globally most of the organizations are using this new technology, artificial intelligence. So there should be the many problems. This requires the thousands of GPUs running continually for weeks or months to train the near. That means for a computation we need a large number of GPUs or CMOS systems in order to compute the large number heavy duty data which AI demands for for running different modules. And the challenge are energy efficiency. The present energy efficiency is the AI is scaling and uses of many more silicon devices. So many researchers are believing to answer. Therefore, alternative computing technology capable of delivering higher performance with a dramatically lower energy consumptions must be explored. Then actually scientists or researchers are doing for the energy efficiency and fast computing devices. So we need to choose another. And then a new computing paradigm. For a decade, the semiconductor industry followed Moore's law. That Moore's law means in each years, the number of transistors becomes double. And then nowadays it becomes the saturation state. So we have to improve the technology, computing technology in the new dimension. Today we are approaching the physical limit. That's the reasons. The major barrier have emerges. First, the power wall. That is increasing transistor density produces the excessive heat. Then today's data centers are producing many significantly high amount of heat that also affect even the climate temperature. And so second, the memory walls. Data transfer between the processor and memory consumes the more energy. Yeah, CMOS systems one process and then data transfer to the memory, and at that time the energy is used. And third is scaling limit. Furthermore, miniaturizations becomes increasingly difficult and expensive. Future AI systems require hardware that operate different from traditional computing architecture. So we need to go the new hardware systems. And let's enter into the Josephson junction. Sorry for interruption. Okay. Josephson junction is there is two layer of superconductor separated by a thin insulator. Superconductor, I think many knows about the superconductor. Superconductor is the material is a low temperature. which shows the zero resistivity behavior, high conductivity, almost infinitely high conductivity, and the basic things of the superconductor is the Cooper pair. When the temperature load and load and load, then the electrons are interact each other to make a Cooper pair. And new types of the entities, that is Cooper pair, which is completely different in behavior rather than electrons. Both of them destroy their behavior, that is fermionic behavior to the bosonic behavior. Cooper pair is purely the bosonic in nature. There is many theories of pairing process. I'm not going to deep about that. Anyway, the Cooper pair forms when the temperature becomes lower than the critical value. The phenomenon is completely different. The thermodynamic phenomenon, electronic phenomenon, other phenomena also completely different in the case of the superconducting nature. And when the Josephson junction based is constructed, the Josephson junctions are various type of Josephson junction can be developed. That may be the planar Josephson junction, annular Josephson junction, even the linear type of Josephson junction. We may call this as a long Josephson junction. In almost all Josephson junction, the difference from one Josephson junction to another Josephson junctions, in whatever way they forms, the behavior are typically different. And in general, the Josephson junction, there are two type of Josephson effect. One is the DC Josephson effect, another is the AC Josephson effect. In DC Josephson effect, the current, we call the Josephson current, flows from one layer to another layer. Even in the zero bias voltage, the current flows from the junction, that is DC Josephson effect. And another is when the constant bias voltage is applied, the AC current will develop. That is another effect which are given in the figure third and fourth. When the Josephson junction, some type of Josephson junction are irradiated by the oscillating magnetic field, then another new types of phenomenon also exist in the Josephson system. That is not displayed here, and the phenomenon is completely non-linear, and the governing equations are different. We call Sine-Gordon equation at that time. I didn't mention here, but all these Josephson junctions they can be used as an electronic device to replace the present electronics devices. Another is Josephson can be model in different way. One is the RCSJ model, that is resistively capacitively shunted junction. Then each Josephson junctions can be equivalent into resistive nature and capacitive nature, as well as the Josephson part. That is typically different from these two. When resistance and capacitor are connected, that is RC circuit we say, and the mathematics or any derivation, any current related is different in case of the Josephson junction. So there is a resistive part, capacitive part, and Josephson effect part are parallel to the capacitor here. And this is actually what we call the RC Josephson junctions. The governing equation is given by IB bias current, that is IC sin theta is the Josephson part, B by R is the current through on the resistance, and C dB by dt is the current crossing the capacitor part. This is actually the model. Its model can be used for the Josephson to describe the Josephson effect. And another advantage of Josephson junctions are Josephson junction can switch in picosecond to attosecond, even less. When we irradiate it by the magnetic field, then the switching mode even can be decreased from attosecond to. Their operating frequencies can reach tens or even 100 per 100 of gigahertz, energy dissipation can be significantly lower than conventional transistor. Why little energy is lost? Little energy is lost as a heat. In a world where energy is consuming is becoming a critical issue, this advantage alone makes a Josephson junction technology is highly attractive. In case of the energy, only this point also very attractive for this. Rapid signal flux transistor logic. Here actually the entities which help us for the to replace the bit, conventional bit. Here actually we use the Q bit. Q bit can be generated in different way. In case of the superconducting systems, there are 4 types of qubit. One is charge qubit, transmon qubit, another is fluxon qubit, and cat qubit too. In case of the fluxon or fluxonium qubit, the flux when in a superconductor, the flux quantized in the superconducting devices. The quantum of flux is fluxon. This is given by S to E, S by 2E. S is the Planck constant, E is the electrons. This is actually the entities which plays the vital role in case to replace the bit to qubit, quantum bit. AI hardware requires massive parallel processing. Modern AI requires massive parallel processing, fast communication, efficient memory access, adaptive learning. These are the required. Actually, nature has solved this problem by making the brain. Our brain is brilliant. It consists of 100 billion of neutron, 100 trillion of synaptic neuron. Neuron as the connector, neuron are the memory processing unit, and synaptic are the connector from one neuron to another neuron. The brain operates only about 20 watt power. This energy efficiency is extraordinary. That is, nature has solved the problem of making the human intelligence. For making human intelligence, the nature has designed our brain using neuron and synaptics like this. So, neuromorphic computing. Neuromorphic computing means we need to devise, we need to develop devices, hardware that works like brain, biological neuron system. Traditional computers separate memory and processing. In case of traditional computer, first process and then store in data in memory, and for next process, data may take from memory and used for processing. That is the separate process, separately done, not at once. But brain does not. Brain do that simultaneously. Neuromorphic systems attempts to mimic the brain behavior, brain-like behavior. The goal is not merely faster computation, but more efficient computation. Faster is better, but first we should prioritize for the efficient computation, precise computation. This paradigm is considered one of the most promising direction for future AI hardware. Josephson junction as artificial neuron. The main advantage are the signals obtained in the Josephson junctions can control. the signal can remain at rest, generate spikes, can oscillate, can exhibit threshold behavior. That in case of the Josephson junction, the fluxion and antifluxion or fluxonium and antifluxonium can superpose at once. That is actually the concept of the quantum computing. In case of the conventional computing system, 0 and 1 are in the separate state. But in case of the quantum computing, 0 level and 1 level can superpose at once. We need such entities which can superpose. In case of the voltage case, voltage low and voltage high case, we cannot superpose the voltage high and voltage low at once. But in case of the qubit, both the situation can be superposed at once, and so we can store more data as compared to the conventional system. Josephson junctions as artificial intelligence. The Josephson junction can be used as synapses and neurons. Synapses determines the strength of connections between the neurons. Superconducting circuit can implement adjustable synaptic weight. Recent Josephson-based synapses exhibit plasticity. Plasticity means once it used, it can be continued for ever in the same state. This is the physical basis of the learning. Therefore, Josephson circuit potentially perform both computation and learning directly in the hardware. Superconducting neural network. Its benefits are ultra-fast operation, massive parallelism, minimal energy consumption. Because of the low resistance, we can have this type of benefit from the superconducting neural network. Superconducting neural network means Josephson junction can be used as a neuron, and the same superconductor can also be used as a synapses by setting the different physically or controlling the thickness and dimension, etc. We can make them as a neuron, as a synapses. This could dramatically accelerate AI application. Recent breakthrough. Research in the area has accelerated significantly over the last few years. Scientists have demonstrated programmable superconducting neurons. That programmable means superconducting like, not exactly superconducting. That is on the basis of the algorithm. They are doing so. Synaptic plastic in memory computing, large scale SFQ, single flux quantum circuit. The recent devices have achieved operational frequencies exceed 40 gigahertz, while consumes extremely small amount of energy. These advantages suggest that the practical superconducting AI hardware may become feasible within the coming decade. That is, we need dive into the Josephson junctions anyway. There are many Josephson-based AI accelerator. Where might these systems be used? Potentially application used scientific computing, where large simulations requires enormous computational power. then all data centers need this. Autonomous systems that require real-time decision making. Real-time decision making means the processing and memorization should be done simultaneously. Otherwise, it takes time. Space explorations where power resolution are limited. So actually, in the space systems, power is does matter, because only the solar power can be used as the electricity in the space systems, I think. Sorry. Another is defense and security applications. Data is very large. As AI systems require ultra low energy consumption, that is almost approach to the brain-like systems. Synergy with quantum computing. Another fascinating aspect is the connection between Josephson junction and quantum computing. Quantum computing is another pioneering technology, pioneering field, which we are using on the basis of the various algorithms like SOR, Grover's, etc. But still, the quantum computing is not has the opportunity for device applicable. Only the algorithm-based quantum computing is going on. Most superconducting quantum computers use Josephson junctions as their fundamental building blocks. There is another way of using qubit in quantum computing, that is photonic qubit, laser qubit. Other qubit also can be generated, but it is not very energy less consumption as compared to the Josephson junction devices. So I wrote, in my titles I wrote ultimate, that is ultimate. In present days, any more research also is going on for the quantum computing with another type of qubit, like I said, photonic, laser qubit. But Josephson junction has 4 types of qubit. I already mentioned that one is charge qubit, transmon qubit, and Foxon qubit. Next one is cat qubit. Cat qubit is hybrid type of qubit. But in case of the Josephson junctions, depending on the dimension, depending on the layer thickness, geometrical parameters, we can design the different type of qubit. We can also transit from one type of qubit to another type of qubit. That is actually the pioneering properties of the Josephson junctions. That is, the qubit depends on the layer thickness, dimensions, etc. And even the nature of the material too. If we use magnesium diboride, then there are the 2 gap, not actually like a band gap, superconducting gap type of, that is ordering. In case of some superconductor, the ordering is one type. In case of another state, the ordering are 1 and 2 types simultaneously. Then 2 gap, 3 gap superconductor can also be developed. And some type of superconductor as topological superconductor, 2-dimensional superconductor also are interesting. So, sorry. these are actually the superconducting behavior. What I am saying that actually most usable qubit is the fluxon, anti-fluxon. These are governed by the Sine-Gordon equation. This can be obtained in the long Josephson junction. In case of the long Josephson junction, there are the fluxon. Fluxon is the qubit for that. The challenge is still, there is still the challenge. The most obvious challenge is cryogenic cooling. In one way, we have the advantages for data processing and fast computing. We have the benefit from one way. On the other hand, there is still the challenge. Either we need to have the room temperature superconductor, or we need to have the cryogenic systems to make a superconducting state of our desired material, which we choose the materials. What materials we choose, depending on that materials, we should cool the materials below critical temperature in order to have the superconducting nature. First of all, we fabricate the devices, and the devices is cooled under the critical temperature, below the critical temperature. Supercomputer requires very low temperatures. So, fabrication technology are also less mature than CMOS manufacture. Then we have to improve the new technology for the fabrication. That is another challenge. It is also still going on how to set up that type of fabrication technology. And new softwares and programs framework must be developed. For the present quantum computing, the Grover Shor algorithm are developed, but when we fabricate the superconductor computation, superconducting network systems, we should develop another type of algorithm. Someone will may come after that in order to modify the present algorithm. A new software and programming framework must be developed. The history of technology shows that revolutionary technology often begin with significant engineering hurdle. So, in one way, there are the many benefits, but in the other way, there is also the challenge mentioned, challenges mentioned like in this slide. Future roadmap. Looking at it, I envision 3 stages. In the near term, that is first decade, I think, hybrid CMOS superconducting systems will emerge. We will not jump directly into the superconducting systems. First, stepwise, we should achieve that ultimate systems. First of all, we should make the hybrid mode, and then in the next decade, I think in the next decades, let me say about 2040, the medium terms, dedicated supercomputing AI accelerators may become commercially available, like the present hardware systems, like present desktop, present hardware systems, present mobile system, present any systems will be replaced by the superconducting systems in the next decade. And then in the longer terms, large scale neuromorphic superconducting processors. Neuromorphic superconducting processors means at that stage all the processors work almost identically with the brain. Biological network systems. Looks like biological network systems. So this is the roadmap. The converging of AI superconductivity and quantum technology may define the next era of computing. Why Josephson junction may become the ultimate AI devices, which I made, according to my title, it is why it is ultimate. There is other devices also which can be used as a computing systems, which can be used as a qubit, quantum computing qubit. As I said, photonic qubit, laser qubit, and other also, we can generate a qubit. But why Josephson junction's qubit is ultimate? So it has a quantum functionality. Other qubit may not be the quantum functionality. Josephson junction is compositively quantum functionality. That a little bit change in the parameters, there is the dramatical change in the entities formation. That is, I said, qubit. Ultra high speed. Its speed is very high. Ultra low power consumption. The power consumption should meet the brain-like power consumption. Only 20 watt is consumed by our brain. We have to develop, if we are going to achieve, we are going to synchronize the brain-like devices, systems, the power consumption should be near about that limit. Ultra low power consumption, that is a natural neuron-like dynamic. Another one is compatible with neuromorphic computing. Neuromorphic computing means any data are processing, how our brain is done by that. Okay, finish. Thank you for your attention. I look forward to your questions and discussions. These are some references. And I conclude my talk saying that the 20th century was built on semiconductor transistor. The 21st century artificial intelligence be powered by Josephson junction. That is, 21st century is the century of Josephson junction. Thank you.
Any queries? Thank you, sir, for giving such a lovely presentation. Thank you, sir. Sir, please come on stage. Sir, you. I humbly request you to, sir. Sir, I humbly requested to you, please come on a stage. Sir, I humbly requested you to please come on a stage. Now I requested.
I'm requesting Professor P.L. Burman, Professor Soto to facilitate with the token of amount as a certificate to Chetri sir.
Now I requested to Dr. Dipender Hamal sir to be come on a stage. Dr. Dipender Hamal sir. Now I requested to Dr. Raju Kunal sir. A big round of applause, guys. Now I invited to Dr. Puneet Kumar Srivastav sir, our next plenary speaker from University of Lucknow in the Department of Physics. Now mic is yours, sir.
Good afternoon. I will be talking to you about quantum plasma, which is the fourth state of matter in the quantum regime. I am coming from Lucknow and I am working in the Department of Physics. This is my department. And this is my lab. This is the work group with me. And basically, when we talk of plasma, we talk of three kinds of plasma. The first one is the classical plasma, of which Langmuir was the pioneer. The second is the dusty plasma, of which Professor Padmakant Shukla was a pioneer. And the third one is the quantum plasma, and Professor Manfredi is the pioneer in this. And I have been fortunate to work with the two legends, Professor Padmakant Shukla, and Manfredi is my collaborator. So when we talk of classical plasma, that's a high temperature, low density plasma. And as the density increases, the degeneracy comes into picture and the plasma becomes quantum. The applications of quantum plasma rise from astrophysical bodies to inertial confinement fusion, the semiconductor plasma. So basically, in the white dwarfs and neutron stars and cores of the giant planets, we find these quantum plasma. In the laboratory system, we have in the laser produced plasmas, in the inertial confinement plasma, in the condensed matter system, in semiconductors, and all the high pressure systems. Apart from these, the gluon plasma, which was generated just after the Big Bang, is also a degenerate quantum plasma. And the applications in the future of such plasmas will be in nanotechnological devices, in photonic devices, in advanced materials, and in the fundamental physics while studying the Big Bang. So basically, the occurrences in the cores of the Jovian planets, the white dwarfs and neutron stars, the quantum electron gas, Coulomb crystals, and all the nanoscale devices in the semiconductors, the spintronics, nanotubes, quantum FEL. When this plasma comes in the influence of the magnetic field, the spin polarization comes into picture, and there are numerous effects. The spin affects these quantum plasmas. So the spin polarized environments are the semiconductor plasma, the laser magnetized nanoplasma, and the spin-odds ones. So basically, when we start to define the quantum plasma, it is the high density plasma, and the it's so much dense that the interparticle distance becomes of the order of the de Broglie wavelength, and the wave functions overlap. Then these quantum effects come into picture. So there are numerous factors, like the statistics which govern these plasmas is the Fermi-Dirac statistics, and the statistical pressure is the Fermi pressure. The particle distribution is the Fermi-Dirac. And this is the book which has been authored by me on quantum plasma. This is a fundamental book, in which I have tackled right from the basic electrodynamics to the frontiers of quantum plasma. This has been published last year by Springer. So why quantum plasma theory is necessary? We have seen the numerous applications. To explain the physics behind this, we need to develop a theory, and this is the primary theoretical framework that is required for such kind of plasmas. There are numerous models, but till now the most prominent one was the quantum hydrodynamic model, which is a classical model converted into quantum one. But since it is not purely quantum and the relativistic effects are not taken into account, the model is classical. So there was a need to develop a quantum model for it, and we went for developing a relativistic model for quantum plasma. So this paper was published last year in the review Letters. So basically we constructed the propagator with the quantum equations, and these were the Euler equations that we came across. You may notice that all the fields are quantized, but we were able to have a quantum system. But one thing you will notice, that we took the electromagnetic wave as a wave, but if we want a system to be purely quantum, so that wave nature should be absent. So we went further and we constructed a new model taking the electromagnetic field, the electromagnetic wave, to be a field coming from a photon. And the mathematics required was not classical, it was a Podolsky photonic field that we took, and we developed this model. So these were the equations. These were the photonic fields. So apart from developing this model, we went for studying the semiconductor physics, from the quantum plasma angle, how the quantum plasma effects govern the semiconductor conductivity, and especially the piezoelectric effects, because all the sensors now that we are using in the nanoelectronics are based on piezoelectric crystals. Secondly, we went for the kappa distribution, like Professor Anil Bhardwaj was talking in the morning about the plasma distribution near the moon. So that's a kappa distribution. So we went for those studies for the kappa distributed quantum plasmas. These were some classical studies that we performed with the QHD model. These are the latest studies. Apart from the theoretical work that we have done with the study of the millets with plasma activated water, and how the fuel enhancement can be done, how the blending of the fuel can be done using a plasma blender. So these are the latest publications which are being published this month only in various journals. And the cross-disciplinary applications are the quantum information, nanophotonics, fusion and acceleration, and biomedicine. Like the earlier talk was on artificial intelligence, so quantum plasmonics will play a crucial role in developing the quantum computers. So these are the emerging paradigms, and there's the quantum thermodynamics, cosmology, gravity, and machine learning. And the questions for the future are we have to go for the non-perturbative dynamics, the quantum classical transition, then the quantum vacuum engineering. That is to be explored. And I end my talk with this, that the quantum plasma defines matter at its fundamental limits, from the origin to the universe to its ultimate destiny. Thank you.
Thank you, sir, for giving us such a lovely and knowledgeful lecture. Now I requested to Professor A.K. Srivastava sir and Professor Solo sir to come on the stage and facilitate to Dr. Puneet Srivastava sir. Now I have a big round of applause for Dr. Puneet Srivastava sir, delivering us such a good lecture on the quantum physics. So now lunch is ready to be. Now the section is getting be ended. Now lunch is ready. No, good for lunch. The online section should also be ended here. The ceremony was about distribution ceremony started at 1 o'clock after the lunch. 1:30.